Thank you.
Oh, already? Well, we'll leave that on. No. Good morning.
Sounds good. Okay, welcome everyone. Thank you to all of our participants, as well as our attendees who got to listen to us do some nuts and bolts work beforehand. I'm so excited to be the presenter today, or the moderator today, for the 2021 UO's Undergraduate Research Symposium session titled Migratory Stories, Sea, Land, and Air. This morning, we will be hearing 4 presentations by students from across disciplines on campus, all of them theming around some sciencey fields, as well as some global perspective into those science fields. Each student and student group will have 10 to 15 minutes to present. We do ask that all attendees be respectful of the presenters and be sure to mute yourself. If you're not muted, Ellen will do her best to mute you yourself in case you accidentally unclick the button, but we want to make sure that the visual and verbal spotlight is on the student presenting. If you have any questions throughout the presentation, there is the Q&A chat box that you can feel free to type your questions into. One thing to note is that as a presenter on Zoom, it can be difficult to both read questions at the same time as presenting. So if your question isn't answered immediately, I will do my best as moderator to make sure they are asked at the end of the presentation. You'll also have a full availability to be able to ask questions at the end of all presentations as well. Just for the funness sake of all of this, I do ask that you ask questions. This is kind of a culminating experience for a lot of our students here. So without further ado, let's get started with our first student presentation today. So Anna Brown is a senior in the biology program. They have been working in collaboration with Alejandro Brambilla and Marissa Lamasse. I apologize if I mispronounced this, but all 3 of those collaborators work in the UO's Hallett Lab on campus, which aims to produce usable science to improve our ecosystem management and restoration. Anna will be presenting their and their co-collaborators' findings in a presentation titled Assessing and Promoting Pollinator Diversity in a Hazelnut Orchard Using Cover Crops. The floor is yours, Anna.
Awesome, thank you. I'm just going to go ahead and share my screen. Hopefully that worked. Okay. Hi, good morning. I'm Anna Brown, and I am a current senior in the Hallett Lab at University of Oregon. And my thesis project is assessing and promoting pollinator diversity in a hazelnut orchard using cover crops. And I'll break down what all of that means over the next couple minutes. I also want to mention that this is current ongoing research, so I'll be collecting data for this project over this spring and summer, and I'll be defending my thesis in fall 2021. So to begin with, what is a cover crop? It's a plant that's grown secondary to a primary agricultural crop. So this might be plants that are grown as ground cover in an orchard or between rows of an agricultural crop. And this is what this experiment is testing. We want to see if we can use native Oregon plants as ground cover in a hazelnut orchard. So in fall 2019, seeds for our cover crops were seeded in 3 hazelnut orchards in Marion County at Laing Farms. And over the next 2 years now, we've been looking at how these plants are growing, reseeding themselves, and resprouting themselves. So the kind of things that we seeded are our native Oregon plants. We have a variety of Willamette Valley prairie species that are flowering. We also seeded a non-native industry seed mix, which is kind of an industry standard and includes things like oats and vetch and clover. And then also what we see in the orchard is weeds. So these are species that we didn't seed purposefully ourselves, but show up anyway. So they are really weedy, and this includes things like dandelion and chickweed. So this leads into the main aspect of what I'm researching, which is pollination on all of these flowers in the orchards. So in this picture, that is a little Ceratina, a small carpenter bee, pollinating Plectritis, which is one of our Willamette Valley prairie species. So hazelnuts themselves are a wind-pollinated crop. So they don't necessarily need pollinating insects to produce nuts, but pollinators can help make sure that our cover crops can reseed themselves and be sustainable. And these flowering cover crops are also kind of a benefit for these insect species in the surrounding landscape. In a hazelnut orchard and in kind of all the farms that surround these orchards, it's pretty monocultural, it's pretty homogeneous landscape. So having kind of floral diversity, a variety of flowers for these insects to feed off of and forage on can be kind of a benefit for their health. So in the Willamette Valley, we have flies, including syrphid flies, beetles, wasps, butterflies, and bees, and all of these act as pollinators. And kind of currently, we don't really know how many species of these insects live here and what their exact ranges are. So this project is also pretty helpful for recording occurrence data and species information and what flowers a lot of these insects prefer, which we don't necessarily know yet. So I'm looking at pollination by two metrics. First one is density. And density is essentially being counted as how many times a particular plant is visited by an insect. So on our flower on the left here, we have 5 visits. It's fairly dense as compared to this flower on the right, we only have 2 visits. It's less dense, less visitation. And the second metric, which I'm measuring pollination by, is diversity. So diversity is a measurement of how many kinds of pollinating insects are seen on a plant species. So on our left flower, pretty diverse. We have 6 different species of insects that are visiting it, as compared to our flower on the right, less diverse, only 3 species seen on this. So I'm counting diversity in the field by netting insects off of flowers, and then I'm able to identify these specimens back in lab using a dissecting scope. So in this first figure, it's kind of a lot, but I'll be showing some of the initial data that I collected last month in April. So this is a diversity figure. This is information about collected insects in the field. So in this figure, I'll just be looking at bees. I did also collect beetles and flies and wasps in April, but bees are a pretty good metric to show for this figure because they're really good pollinator because they deliberately collect pollen off of flowers to provision their larvae with. So on the left side, there are 5 bee genera that were collected in April, and I have photos for all of those bees on the far left. And then the lines in between connect the bee to the plant that they were collected off of. So Geum, Lomatium, and Plectritis, those top 3 flowers are all native Oregon species, and everything else below that is weed species, which again, were not intentionally seeded. So there aren't actually too many plants that we intentionally seeded out of the native species that are flowering yet. There are 15 in total, and we only have 3 flowering in April, which is pretty early. We're expecting to see probably peak bloom around June when most of the species will be flowering. So what is interesting to note in this figure is that weeds only supported 2 out of the 5 bee genera that we saw during April, whereas the native flowering species hosted all 5 genera. So here I also have some of the data about insect density. So the way that density is collected in the field is I set a timer for 2 minutes in kind of a 1 meter by 1 meter plot of where our cover crops are growing and flowering. And I tally the number of times an insect visits a flower, recording both the species of flower and the kind of insect that visited it. And so I have a morphospecies category instead of a genera or a species of insect because it's really hard, if not impossible, to determine an insect to species or genus level by just looking at it on a flower for a couple seconds when it lands if you're not actually collecting it. So instead, I use these morphospecies categories. So combined, I actually saw in April about as many total insect visits to native plants as there were to weeds, even though there are more weeds flowering in April than there are native plants. Also kind of interesting to note, the weeds supported 5 morphospecies categories and the native plants had 6. And again, the native plants on the bottom are geum, lomatium, and flectritis. Everything else is a not purposefully seeded weed. So just based on April, there isn't really enough data yet to draw firm conclusions about which plant species are better at supporting pollinator density and diversity. But I'm hypothesizing that the data that we collect from April all the way until August is going to show that we'll see higher insect diversity on native plants than the industry mix or on weeds. And I'm also expecting to see that we'll see higher insect visitation on those native plants than on the industry mix or weeds. And that will apply across all of those morphospecies categories. So one other factor that's going to shape how these results are ultimately going to be implemented is by looking at what plants can survive in a hazelnut orchard in the first place. So a plant might host a lot of insect diversity and be really frequently visited, but it doesn't matter how popular a plant is if they can't survive in an orchard in the first place. So these plants are going to go through basically a filter here. On the first level, looking at environmental factors. In a hazelnut orchard, the hazelnuts leaf out in the spring. So if plants can't survive being shaded, they're out. They're going to get filtered out. This on this level also we'll be looking at competition. So if plants can't compete with neighboring cover crops, then they're not going to make it past this level essentially. So then on our next level, we'll look at mechanical disturbance. In a hazelnut orchard, there's tractors driving around, and also in fall, all of the vegetative growth needs to be cut back because in fall, the orchard floor needs to be smooth so that they can collect hazelnuts off of the orchard floor. So if plants by the fall either can't regrow themselves or haven't gone to seed yet, they're not going to be able to survive in the next season. It's not super sustainable to have to reseed your cover crop mix every single year. So if they can't sustain themselves at this level, they're kind of out. And then our last level of the filter is going to be looking at what ecosystem services these plants can survive or provide. So I'm looking at one aspect of this, which is the pollinator promotion, but other ecosystem services we're looking at in the Hallett lab are moisture retention and erosion control. So at the end, we're going to end up with hopefully an ideal seed mix. So the main goals of this project are to have a cover crop seed mix that can just grow in a hazelnut orchard really well, while maximizing pollinator density and diversity. And also we want it to provide the most benefit and sustainability to hazelnut farmers by offering them things like erosion control and moisture retention in the orchards. And that concludes my my presentation. Thank you for attending and listening. And thank you to Alejandro and Marissa for collaborating on this project in the Hallett lab. I'm happy to take questions and I'm excited to see the other presenters on this panel.
Great job, Anna. For anyone who has a direct question follow-up right now, feel free to type it again into the question and answers chat. You can also unmute yourself, I believe, and ask your question directly.
I have a question. 'Cause I noticed on you, it looks like, are you focusing on any specific pollinators, like native versus non-native pollinators? I know honeybees were one of the big ones you looked at, but they're not native to the Pacific Northwest, or just looking at pollinators overall?
Yeah, that's a good question. We're looking at things overall. I haven't been collecting honeybees because they're pretty easy to actually identify by sight. And also I haven't been collecting them because they're introduced to the landscape. So right by one of the hazelnut orchards, they have a marionberry farm. So they set up like 5 or 10 hives there. So it's really hard to tell how many would be like visiting in this environment otherwise, because they're all introduced every year. But yeah, we are looking at both non-native and native because we just want to see what is visiting the flowers and what has the ability to make sure that they produce seed right now. And I think that's definitely something that I'll be discussing in my final thesis about whether we're seeing more native or non-native insects on especially the native plants.
And could you remind me, if you've already mentioned, when are you planning to have your final thesis finalized?
I'll have it finalized and I'll be defending it in fall 2021. So at the end of this year.
Okay, so a couple more months of research, it sounds like.
Yep.
Very cool. Any other questions for Anna before we move on to our next presentation? So in the chat, there's a question. Are there some weed species that are really good pollinators? For example, are there a lot of pollinators on weeds because a lot of weed species, because of weed species that help pollinators?
That's a really good question. So I couldn't tell you the exact like nutritional statistics of all of our weed species because yeah, there are about 4 or 5 out there. But I will say, in general, the way that I view weeds, and I think the nutritional benefit of them is pretty low. The reason they're getting visited a lot in April is just because like there essentially isn't that much else to visit. So if there are bees or flies out that are foraging for nectar and pollen, like there's very little else to go to. Although this is going to be something that we'll explore as the months go on and we start to see more of our native species flower. I did do some initial data collection last summer and it wasn't really what we saw, but if, for example, we see more insects are visiting the weeds than our native plants, that would definitely be something to explore and start looking into like nutritional content of the weeds. But what I've seen in the past and what I'm kind of expecting to see is that when there's just more things available, the weeds are like going to be less popular because insects will have kind of other options. It won't be the only thing that's flowering.
Thank you. We do have another question as well from Lina, something I was thinking as someone who is a little nervous around bees and pollinators, so bugs of any kind, but how do you catch pollinators?
Yeah, I think I mentioned this in my diversity figure, but I just do targeted netting. So with a net, aim for a bee or an insect that you see on a flower and catch it and collect it. I've only gotten stung once this season so far after collecting probably like over 100 things. So I'm not super worried about it. But you can either like, when you're getting an insect out of a net, you can just kind of hold a collecting tube or a vial up to it. So you don't even have to like touch it or anything. Yeah, it is not a painful process to collect insects.
And once you collect them, where do you store them or help them live?
So collected insects, unfortunately, do not live. I put them in the freezer overnight and then they get pinned so that I can identify them. It is kind of a casualty of doing studies that involve insect identification because there really is not an easy way to effectively identify insects to a species level without actually like collecting them and pinning them so you can look at it under a microscope, because some of the features are just so small. You really do have to look at it on like a magnified level. Although I will say by doing this like targeted netting rather than putting out for bees, you can do things like collecting bowls, which are just like bowls of water with like soap in them so that you don't get like surface tension. I will say those are a little bit more general and invasive, like you just collect a lot more insects. So I'm not taking as many insects, like I'm not killing as many insects from the environment through this method as I would otherwise with like passive sampling methods like the bowls or putting up like sticky traps or something.
And it sounds like your work is also helping with eventually, you know, building insect population as well. So that makes sense.
Yeah,
Great.
yeah, I definitely think so. I've already been kind of checking insect records for the Willamette Valley, and I'm pretty excited about what we've collected because we're going to be able to add some more species and genera occurrences so we get kind of a better picture of where things are located in the Willamette Valley, where different insects live ultimately, which is kind of cool.
Very cool. Awesome. Well, I learned a lot, and I definitely have some questions that we can maybe save for the end, and some of our other attendees might as well. But if it's okay, I would like to move on to our next speaker. So next we have Yalin Li, a senior majoring in environmental science with a minor in biology. She has been collaborating with biology associate professor Dr. Kelly Sutherland and a biology graduate student, Marco Corrales-Ugalde, to put together this research project titled "Do Seasonal Changes Affect the Nutritional State of Jellyfish Along the Pacific Northwest?" Yalin discovered this lab when participating in the Students of Color Opportunities for Research Enrichment her freshman year at the U of O. Outside of this work, Yalin is also serving as the events coordinator for affiliated students for undergraduate research and engagement. So she helped coordinate this very symposium and possibly even this session. So Yalin, we want to welcome you, and we are excited to hear about your work around the nutritional state of jellyfish.
Thank you. Let me see, get my screen share. And it, hope you guys are seeing the right screen. I'm sorry, I have dual monitors, so it gets a little confusing sometimes.
You're good, we can see it.
Okay. All right, so hello, I'm Yalin. I'm currently a senior and I'm studying Dr. Kelly Sutherland's lab, and my research topic is going to be do the seasonal changes affect the nutritional state of jellyfish on the Pacific Northwest. Let's see. Oops. Okay, I want to start talking about our research goals and questions because there kind of is like 3 parts of this study. So our main study is kind of looking at does the gonadal index, which is the gonad area provided by the bell diameter, vary in hydrogen reduced between winter and summer. The reason why we're looking at gonadal index specifically is that this is so we can look at the proportion of the gonad area in proportion to the bell area. Because we're going to assume that the gonad area and bell area can be a little bit higher between winter and summer, because summer's going to have a little bit higher productivity, so it's going to be higher in summer compared to winter. But by looking at the gonadal index, we can actually see if there is a little bit like prioritization towards reproduction over just overall somatic growth. And so kind of like our sub-studies that we looked at, does the growth of hydrogen vary with different food levels? In doing so, we did a feeding/starvation experiment where we caught live hydrogen reducing, and we just kind of fed them different levels and see if that caused variations in their growth. And the final part of our experiment is we looked at creating a correction factor to estimate the live measurements from preserved samples and for the nutritional state in the natural environment, because the ones that we took for this first experiment were actually already preserved. We took them from winter and summer 2018, 2019. And since we wanted to look at how they were in the natural environment, we created correction factors to kind of estimate from these preserved samples. And so just a little bit of background. The reason why we're looking at during the Pacific, along the West Coast, there's a process called upwelling during the summer month, which is where wind blows across like the surface of the water, and it kind of just offsets the water along the coast. And in doing so, some water rises deep below that's very rich in nutrients, and this causes an increase in primary productivity. And this in turn changes the composition of zooplankton communities. And how this relates to jellyfish is that they predate on these zooplankton communities. And there's a lot of different things that are considered plankton, like pelagic fish, eggs, and also as crustaceans, which are very high commercial fish that we feed on. And since recently, due to a lot of different environmental reasons, there have been a lot of jellyfish blooms, which might mean that they're going to predate a little bit more on these zooplankton. And so in past studies, some people have found that when food is a little bit limiting, jellyfish will actually focus more on their reproductive growth over their somatic growth to help increase their offspring, because most organisms, their overall goal is just to reproduce. And this is kind of why we were looking at gonadal growth area over bell diameter instead of just overall. So then we actually see if they really are prioritizing reproductive efforts over somatic growth. On the other hand, when food is abundant, they'll focus a little bit more on their somatic growth. And just some studies we're using as a baseline. Is this study by Vichy and Basal in 1998, where they looked at Scaphonusia aurelia, which might be those like really pretty like purple moon jellyfish you might see in the zoo. And they saw that when they are very well fed, they will increase their bell diameter compared to when they are starved, they'll decrease their bell diameter. So you can see how this kind of variation in food levels changes their growth. And then we talked about earlier that reason why we use formalin and why we're using that correction factor, said there's a lot of caveats in formalin. It's really great because it's very well known, everyone uses it and it's widely available. However, it does cause changes in the volume of the jellyfish. So you can see there's a decrease in the height, diameter, and the weight. So that's why we want to create these correction factors so we can estimate their live samples. And so I'm just going to give a little introduction to the two organisms that I looked at specifically, which are Clitogregaria and Eutenata indicans. These are both hydrozoans and within the phylum Cyanidarea, and they're both very small. As you can see here, Clea is only 5 millimeters, like in the radius, and about 10 millimeters for this one. And so you see they're very small, so they're actually not very well studied, even though they're very highly abundant. The reason why we chose them because they're highly abundant pretty much year round, especially Clitogregaria, which allows us to kind of see how they change throughout the seasons. So just some of the methods that we use. Most of the organisms that we collected were from the North California current, specifically along the Newport line and the Trinidad line. The ones that we took from starvation preservation experiments were taken from Charleston, off the coast of Charleston. And we use a couple different methods collection. So this is called a mock nest, and that's how we collected the Hydromedusae from the Newport lines to the Trinidad lines during the winter and summer of 2018-2019. And to collect the plankton for the starvation experiments, we used this process called the ring net. And then for some of the jellyfish we collected, we used dip netting, where you just kind of see, when you see jellyfish float by, you just push the little thing under and the backwash of the water will flush the jellyfish into the container. And so for our starvation feeding experiments, we kept the jellyfish in seawater in these 100,000 milliliter containers on a sea table to kind of help regulate the temperature. And they were separated into 3 groups. Ones that were fed twice a day, once a day, and unfed. And so how we fed them is that we had this plankton solution that was just plankton mixed in seawater, and then I would just mix it up, make sure that's homogeneous, and then I would feed them 10 milliliters to the ones that were fed. So the ones that were fed twice a day got 20 milliliters overall. The ones that were fed once a day got 10 milliliters overall. Ones that were unfed were not fed at all. They were just there. And then this lasted for about a period of 1 week, which seems very short, but the Medusae are only known to live about 3 months. So 1 week out of 3 months is a good portion of their lifetime. And how I looked at their morphology, so I just basically took a ton of photos using a microscope and a camera attached to it. And so for starvation experiments, I just took photos right before the experiment started and at the end of the week. For the preservations, I took them right before preserving and then intermediately over time. So like 2 days after preservation, 4 days, 5 days, so on and so forth. And then for the ones for winter and summer, we already had those samples that were previously preserved. So I just took them and took them as is. And I analyzed the pictures using IMSJ. And for bell diameter, I just took a straight across, whatever I could find was the best estimate of the bell diameter. And then for the gonad area, I traced each individual gonad a little bit more clearly than what I did here, and then add them all together for overall gonad area. So then here's some results. So for the starvation experiment, contrary to what we thought we were going to see, where the ones that were fed more, we'll see increase in diameter, and ones that were being a little bit starved, you see a increase in gonad area. We actually saw that all of them decrease in bell diameter and gonad area, which could be due to a lot of different reasons, because one, we found that maybe we just didn't feed them enough. So all of them were basically just starved, and then also there wasn't any water circulation. So they might not have encountered their prey. But nevertheless, we did see that the ones that were completely unfed did decrease the most, especially in their gonad area. And this might be because we have seen that some jellyfish can kind of do a process of like reabsorption. So when they have no food at all, they will actually reabsorb their gonads. And sometimes they will even disappear completely. And then for our biomass loss formulas, I didn't show the formulas here because they're not really that important for you to know. But we did see from these experiments, because we actually did take the bell diameters and gonad areas, but we also did weigh the bells and the gonads separately. We dissected a subsample of them. As you can see here in the bell, in this bottom left one, you can see that they decreased a lot more than they did in the gonad, which shows us that the bell changes a lot more than the gonad over time, which will be important for the next slide. So you can see here for the uncorrected data, compared to the corrected data, if you look at the y-axis, the proportions are drastically different. Because once we corrected the factors, the bell grew so much more than the gonads that overall for all the organisms, the live samples, the gonad was smaller than in proportion to the bell in the corrected versus the uncorrected. But as you can see, the results did kind of show what we kind of expected it to, where during the winter, the gonad index or the proportion will be larger in the winter compared to the summer, as you can see in the green, blue, and the red, which shows us that during the winter, there is a slightly larger gonad area in proportion to the bell area than there is in the summer. And just to kind of like go on a baseline why we kind of use the gonad area compared to just bell gonad area in general. So you can see here during the summer, the bell diameter and the gonad area is larger in both species than the summer and the winter. But you can also see there's much more of a difference between the bell diameter than the gonad area between the winter and summer. So then you saw that the bell does grow more in the summer than the gonad area does. So just some of our conclusions that we have found is that there are some variations in food availability. It does affect the growth of hydromedusae. And then the bell does change a little bit more drastically in formula than the gonads do. And that there is a difference in the bell and gonad growth due to the difference in nutritional state, the potency of prey. However, we found Marco, who's one of the graduate students working on me, he looked at kind of their gut contents in the jellyfish, and he found that they actually eat about the same amount in winter and summer. So when we looked a little bit deeply into this, we can see that it might not be the density, like the amount of prey that's available for them, but that's the nutritional value of the prey that they can find. And so it's been found that during the summer, their copepods are one of like the main thing that the hydromedusae feed on. And during the summer, northern copepods are more dominant, while in the winter, southern copepods are more dominant. And these northern copepods, which are dominant during the summer, are actually more nutrient and fatty acid dense than the ones during the winter. And so it might not be like a factor of how much food is available for them, but like how nutritious that food is. And just to kind of wrap up on why this study is important is that recently, due to like a lot of different environmental factors, especially eutrophication, there's been a lot of jellyfish blooms, especially in non-local areas. And jellyfish have been known to kind of like decimate fish due to kind of like they feed on fish larvae and eggs, as well as a lot of crustacean larvae. So helping us understand like the dynamics of these gelatinous predators will help us better understand like their effects on plankton communities in the future. That's my presentation.
Great job. So any questions for Yalin?
Yeah, I just had a clarifying question. I know that you touched on this in your presentation, but what was your reasoning for why you think the bell changes more drastically?
I think we don't really, because I'm not a chemist, I think it has to do with kind of like the tissue density. Hydromedusae, they're, like you can see, they're very clear. So there's probably just a lot of water matter in the bell compared to the gonads, which might be just a little bit more like, not the tissue dense. So there's not like just like a lot of, so like the tissue in the bell probably just got like the water content just got drained a little bit more than the gonads did. Because the gonads kind of got more fixed, like they kind of stayed the same throughout fixation, while the bell kind of started to like shrink over time. And like, I'm not a chemist, so I can't really tell you, but I think I'm pretty sure it has something to do with like the water content in the tissue that's available in the bell compared to the gonads.
Okay, that makes sense. Thank you.
I had a question kind of about, I guess, historical records and how they compare to, you mentioned that there are jellyfish blooms that have happened in recent years. I was wondering if you have any like comparisons on historical populations versus now, if you're noticing like major changes in populations of the medusae.
I'm not sure specifically in just the medusae. I know there has been quite a few, like especially along coastal areas, where like jellyfish blooms are kind of just like sprouting out. Because you know, just kind of like looking at environmental factors, there's been a lot of different like gelatinous organisms that have been spreading out, kind of like pyrosomes. I don't know if you know what pyrosomes are. They're kind of just spreading out like randomly. And so just like in general, a lot of like gelatinous species are kind of just been blooming. And I think that they're kind of seeing this might be due to kind of the warming of the water, and as well as since a lot of them prey on plankton, and since there's this increase in plankton and primary production, they also might be following that current.
Sorry, can I ask a quick follow-up question on that as well? Are you expecting to see that be kind of self-reinforcing almost, like there are bigger populations that can reproduce more and then there's more plankton available in future years as well? Like, do you think populations you'll continue to just see these blooms over time, or will there be like a point where, I don't know, I guess populations will not be able to sustain themselves or fall off due to lack of plankton or environmental conditions?
Yeah, some of them have been seen that there is a little bit of like a population density factor that helps regulate these populations, because at some point they will start competing for food, and when they start competing, they're going to reproduce less. So at every organism has like that forgot I haven't taken biology like 213 in a while, but that kind of tipping point. And once they reach that tipping point and there's too much of a population density, they will kind of just stagnate, and there will be eventually like an end to how much blooms they have, because there is like a population density factor that regulates them.
Thank you.
Great. I think we have time for one more question if anyone has one. Otherwise, we can move on to our next group presentation. Okay, let's move on to our next presentation, which I mentioned is a group presentation with 5 students who have all been looking into the migratory patterns of birds. So I can't go into as much detail about all of the awesome things these students are doing just for time's sake, because it is a larger group. But we have Natalie Lehrbach, a senior in the Global Studies program with a minor in Environmental Studies and Spanish. We also have Rachael Maloney, who's a junior in the Environmental Studies program. Wally McAllister, a senior in the Environmental Studies program, also with majors in what's called 3 PM here, Public Policy and Project Management, as well as an Earth Science minor. Olivia Zajac is joining these students and is a junior in Environmental Studies. And Eloise Navarro is a second-year student who's collaborated on this program, who's a Global Studies and Romance Language Studies major, as well as having minors in both Environmental Studies and Latin American Studies. Without further ado, let's hear on our presentation titled Bird Nerds, Uniting Elementary School Students in Oregon and Mexico Through Shared Migratory Birds.
Awesome, thank you so much for that introduction. I will go ahead and share my screen. All right. Okay, so welcome everyone. Thanks so much for being here. You could call us the Bird Nerds, or we're the Aves Compartidas team. And we're here, we're a group within the Environmental Leadership Program at UO. My name is Eloise, and before continuing, I would just like to make a quick acknowledgement that we're on Kalapuya land, and this is a super important thing to acknowledge, especially within environmental studies work, but for all disciplines as well. So I'll go over our agenda. First, we'll be doing just a quick overview about the project, and then we'll talk about what we've accomplished these past couple terms. Then we'll move into some recommendations moving forward, and then finally some thoughts and reflections.
All right, so I'm Rachael, and I'm going to be giving a quick overview of who we are, who we've been working with, and what our goals have been in doing all of this. So we are 11 students in the Environmental Leadership Program, and so we've been working on a project called Aves Compartidas, which was originally launched by the Willamette Laha Twinning Partnership. So if you look on the left there, you can see all of us along with our incredible GE, Katie Russell. And then on the right, there's a map, and there's Eugene, Oregon in the Willamette Valley, as well as Guanajuato, Mexico in the Laha Valley. And there are birds that migrate in between these two watersheds. And so the Aves Compartidas program partners with elementary schools in these valleys in order to teach students about those birds. And the goal of this is to bring students in Oregon and Guanajuato together to foster ecological and cultural connections through studying our shared migratory birds. And our role as the environmental leadership program in this has been working with River Road Elementary School in Eugene in order to teach students a curriculum that we have developed about these birds, and we've also translated it and shared it with our partners in Guanajuato. So a ton of this project is based in collaborating with our community partners. And a little bit more on them, there's a Willamette Laha Twinning Partnership, and that partnership was established in 2015, and they launched the Aves Compartidas program during the 2018 and 2019 school year. However, we did not become involved as the environmental leadership program until this school year, when we began developing curriculum and teaching it to the students at River Road Elementary School in order to help the Twinning Partnership achieve their goals, you could say. So what are these goals, you might be wondering? Great question. The mission of Aves Compartidas is to unite youth, educators, habitat restoration practitioners, and the birding community for deep cultural connections and sustained conservation of our shared migratory birds.
Awesome. Thanks, Rachel. So I'm Olivia. I'm going to go over our accomplishments. So what did we do? And the methods and outcomes of some of the data we tried to collect through this. So what we did in winter term is we learned theories and concepts regarding environmental education. We developed a 7-week curriculum for 3rd, 4th, and 5th graders. So that was a total of 21 ecological lessons to be taught by us in spring term, and also to be used by the partners in Guanajuato and for future ELP students in this project. And we also created an evaluation and assessment plan, which was how we plan to collect data on reaching the learning outcomes and goals of the lessons. And then this spring, we continue to develop lessons and revise all of our teaching materials. And then we taught the lessons. So our entire team broke into pairs and took on teaching these lessons on Monday, Wednesdays, and Fridays. And so in total, we taught 17 lessons to 42 students at River Road Elementary. And we completed 10 hours of community service through teaching 30-minute lessons for 7 weeks. And following each lesson, we did make sure to have team debriefs in order to receive feedback so we can improve as instructors each week. And then we also worked within individual roles to accomplish much more behind the scenes. So for example, our translation team was a group of 4 of the students on our team who spent the term translating each lesson into Spanish. So very cool. And then one of our teaching materials was a field journal that we created for the students. So this was printed and deposited to the school and to be given to each student. And so the field journals had activities that promoted students practicing being scientists and activities pertaining to each lesson. And we also developed an interactive ecological bingo game that lived in their field journals. So this was to really try to engage students outside of class. And the one displayed here is the 3rd grade bingo activity. And so we also promoted environmental stewardship throughout the curriculum by offering what we called eco challenges in each lesson. And then the method we used was a Google form survey that was filled out by an observer during and after each lesson. And so we included attendance, participation, opportunities for assessment, and overall student understanding in the Google form. And we wanted to collect this data to gain insight on student participation and really attempt to discover changes in knowledge throughout all of the lessons. And so this allowed us to discover how we can improve the lessons for future use and find how student engagement and learning can improve as well.
Cool. Thanks, Olivia. I'm Wally again, and I'm going to go over our short-term outcomes. So we wanted to show this data. This is any changes in knowledge and skills by the students, and this is measured by discovering if the learning objectives are met by the end of the lesson. And so here in this graph, we documented the findings of the average percentage of students that demonstrated understanding of the material. And these numbers all range above 50–60%, which we're really proud of. And so now I'm going to transition to talk about what went well and what could be improved. so that future students in the Aves Compartidas program can build upon our work and continue to help elementary students learn about birds. So students spent time outdoors discovering the environment by actively engaging in the weekly bingo activities and eco challenges. And this meant searching for things like pollinators and different bird species to promote ecological awareness and environmental stewardship. Students experienced multiple types of learning styles through things like watching videos, making observations, and doing coloring activities in their field journals. And they also demonstrated an understanding of the material by filling out those field journals and participating in class discussions. And most importantly, they had fun, and so did we. And so what can be improved for future lessons and future environmental leadership program team? We had difficulties virtually contacting teachers and building rapport with students, but we took it as an opportunity to adapt to an ever-changing world. For future environmental leadership programs, we want to improve collaboration with teachers for a more complete evaluation plan. We want to understand what students already know so that we aren't repetitive in our teaching. And we want to get a better grasp on how to use the students' field journals as tools of evaluation rather than something the students keep to themselves.
Hi, I'm Natalie, and I'm going to be talking a little bit about the lessons learned and an academic reflection from our team. So within our coursework, our team completed teaching evaluations and reflected on what we learned from our lessons. And some of these are that hearing the students share about their experiences engaging with the environment and observing birds is really awesome. And sometimes that takes time away from every single little thing that the lesson plan wanted to achieve. So therefore, we prioritize creating genuine connections with the students over following the lesson plan perfectly. Next, we encourage students to think critically about the environment and how humans relate to their environment, and that's more important than having them memorize and then regurgitate specific facts about the birds. And this yields a deeper understanding and connection to their environment that the students can take with them to their future. And throughout the curriculum, we reinforce the concept that there are many different ways of learning or understanding nature, and these include indigenous wisdom, natural science, storytelling, to name a few, and these are all critical, holistic approaches to shaping our next generation of environmental stewards when used together cohesively. Lastly, we believe it's important to encourage students to make observations. Scientists rely on tools of observation and forming hypotheses to address all the questions that we have about our natural world, and by doing this within our lessons, our students were scientists too. So next, we're going to move on to an academic reflection. So the students weren't the only ones who grew throughout this project. Our team gained communication skills through meeting twice a week and working as a team. We each worked on our own projects, so each person participated and led or co-led a project within our program, such as the translation team or the evaluation and assessment, which helped us gain leadership skills. We developed a successful curriculum with 17 lesson plans about the scientific and ecological aspects of these migratory birds. And building on that, we also gained technical skills, such as the development of successful slideshows with videos and photos. We demonstrated resilience by continuing to push forward in our virtual format despite any setbacks that we faced. And as a team, we gained personal growth and confidence and developed a deep love and appreciation for education. So some acknowledgements. We would like to thank all of our awesome community partners and those who have helped us along the way with this project. And this includes the Willamette Laha Twinning Project, our guiding faculty and project manager, Dr. Katie Lynch and Katie Russell, the River Road Elementary School teachers that we worked with, and of course their students. And this project was a collaboration, so we couldn't have completed it without our current and past environmental leadership Aves Compartidas team members. So thank you all.
Awesome. Thank you so much for listening to our presentation. And I think now we're happy to open it up to any questions. And I will stop the screen share.
Great job, guys. Yes, time for questions. And I do have one just to kick us off. there was a diagram with the learnings outcomes from the students week by week, and it looked like during week 3 and 4 there might have been a dip for all of the students and their retention of what they were learning. Was there something that happened during that week, and if so, how did your team adapt to kind of progress forward and continue an upward trend?
Yeah, I can take that. So actually, during that week, the 3rd week, they had outdoor school, so we missed a week. There were some students, and so we skipped a week, and we were thinking that maybe it was because that the students were at outdoor school and had spent a bunch of time outside, that they were a little drained and maybe not prepared for that lesson. And so that was a something that could have set it off, but yeah, not entirely sure.
Thank you. Any other questions for our group before we move on to our last presentation?
Yeah, I had a question. I was wondering, I think you used 3rd through 5th grade. You taught, yeah, that age level. I was wondering if that was like a specific choice to teach to that age group, or if in the future you would like expand it to different age levels as well.
I can take that question. Yeah, I think that was decided by our partners. And so we didn't necessarily, as the ELP group, decide what the grade levels were going to be, but I believe that we hope to expand it into teaching all grade levels in elementary students, so K through 5th, and it be very similar to a different program in ELP, Restoring Connections, where they can grow upon the knowledge they learn from kindergarten up through 5th grade, and then just build on that ecological awareness throughout elementary school.
That sounds awesome. This is a really cool project.
Thank you.
I'd also like to add on to that I think with regards to like expanding the project as well, I think there's a lot of prioritization and finding ways to make it more collaborative with the partners with regards to the students, and the students being able to collaborate a bit more with the students in Guanajuato. And so I think that's also something that the project will hopefully be striving to do more in future years.
Great. Well, just for time's sake, I want to make sure we have enough time for all of our questions at the end. So we are going to move on to our next speaker and bounce back to you when we have questions later. So next we have Rachael Peterson, who is a senior in the environmental science program with minors in both biology and planning, public policy, and management. They have been mentored by Earth Sciences Associate Professor Dave Sutherland and PhD candidate Nicole Abib in Dr. Sutherland's Ocean and Ice Lab. Their research title includes some words that I don't know how to say, so Rachael, if you could do a full introduction of your title as well, because I do not want to mess up the pronunciation of what look to be potentially Norwegian words.
Yeah, wonderful. Thank you so much for the introduction. I'll just go ahead and share my screen. Okay, so my name is Rachael. Actually, let's wait for this to load. Can you guys all see that? Okay, great. My name's Rachael. I've spent the past year researching the influence of ephemeral ice melange on water properties at Rink Isbrae Fjord in Greenland. So first, why we're interested in studying the ice sheet of Greenland is because of how rapidly it's been losing mass. And part of this presents concern for sea level rise and the evolution of the ice sheet in general. And understanding ocean and ice interactions and how they influence this ice mass balance is important. And one part of this puzzle is ice melange, and this has remained largely unconstrained. So you can think of ice melange as a slushy of icebergs and sea ice, which form at a glacier's termini. So if you look at this picture, this is a glacier. This is its termini, and then here's all of the ice melange. And ice melange has a unique role in the glacier system because it's been shown to exert backstress against the glacier. And this can prevent, or this can slow it down, which is important because when a glacier speeds up, it starts to destabilize itself. And that's when you get those big chunks of ice that start calving off of the glacier's end. So ice melange can be either temporary or persistent, and this varies both spatially and temporally. So for Rink Isbrae, which is on the west coast of Greenland, you'll get a persistent ice melange in the winter, but then come summer and into autumn, this becomes more temporary or ephemeral. So to get an idea of just how temporary ice melange can be, this is time-lapse imagery of the ice melange event at the focus of my study. So on the 3rd, you can see that the fjord is completely empty. By the 6th, ice melange has formed in full force. And then in 2 days, on the 8th, it's completely gone. So to get a scale and a sense of size, this ice melange was estimated to be about 250,000 meters cubed worth of ice. So just the sheer volume that's disappearing in the fjord in just a handful of days suggests that we should be seeing a noticeable impact to fjord water properties. And this is exactly why we're so interested in researching what it's doing. So why changes in the water column are important is because of how they might influence buoyancy-driven circulation. So buoyancy-driven circulation is invigorated by density changes in the water column. So in a fjord or the ocean in general, you'll get water masses stratified by density. The densest layer is going to be here at the bottom, of course, and then the least dense water is going to be at the surface. So in the case of a Greenland fjord, your densest layer is this Atlantic Ocean layer. And then your least dense layer is the freshwater melt from the glacier. So when something disrupts the stratification of these water masses, you'll get circulation. And one particular type of disruption are freshwater fluxes. So that's glacier melt, iceberg melt, or runoff. One thing that it also could be is ice melange melt, but we don't know yet. So why this is important and what ends up happening is you'll get this outflow current from these freshwater inputs. And that generates this compensatory inflow of your Atlantic water layer. And this is important because the Atlantic Ocean layer is far warmer than anything else in the glacier fjord. So what ends up happening is you get this heat flux at depth, which leads to glacier melt and then essentially ice sheet destabilization. What's also interesting about this is because this current of freshwater inputs at the surface is causing this inflow of heat at depth, you're getting even more melting, which could lead to greater rates of this meltwater at the surface, leading to even greater rates of heat flux at at depth. So there's this potential for a feedback loop. Currently, it's completely unknown, like I touched on before, how ice melange fits into this conversation. We don't yet understand how it affects buoyancy-driven circulation, and we don't yet understand how it influences heat flux to the glacier termini. And the biggest reason for this is, as you can imagine, it's relatively difficult to get a ship into a fjord that's chock full of ice. And therefore, no ship, you can't really get these field data input or data points from the fjord. And that's exactly why the data that I'm working with is so special. So in 2013 to 2015, a handful of scientists went out into Rink Isbrae and gathered CTD transects or CTD casts along different transects, and that's shown here in purple. CTD stands for conductivity, temperature, and depth. So what you're measuring is salinity and temperature with depth. So for my study, I'm focusing on this transect right here, which is closest to this glacier termini. The reason for that is CTD casts were taken here on August 4th, and then again 2 days later on August 10th, or 2 days after the ice melange breakup on August 10th. And what this is able to do is capture the first, to my knowledge, instance of in situ fjord water properties, both before and after an ice melange event. So with this data, we can analyze what ice melange is doing to fjord water properties. So here's what we found. So this is, on the left is a plot of temperature with depth. Before is in black, after is in blue, and on the right we have salinity with depth. So what we see is some of the coldest temperatures occurring around 50 meters, and that continues down to 250 meters. And then on the right, you have a freshening at the surface that continues also ever so slightly down to 250 meters. So for my analysis, I chose to focus in on 50 to 100 meters. This is because, again, we're seeing some of the coldest temperatures around 50, but then also we know that icebergs in an ice melange reach up to about 100 meters of depth. In addition, the surface layer tends to be highly variable. So for these reasons, we would expect not only 50 to 100 meters to feel the greatest impact of any melting, but to also be the most reliable data. And so what we end up seeing is that in this area, the water cools by an average of 0.34 degrees Celsius and freshens by an average of 0.18 PSUs. So this is cool and exciting and what we would expect to see if ice melange was melting in the fjord. But what we have to do next is understand if this can really be attributed to ice melange melt, or perhaps it's just another freshwater flux into the fjord during the same time period. And the way we can determine whether this is ice melange melt or not is through analyzing the ocean properties in temperature and salinity space. So this allows us to discriminate between two specific water masses, runoff or submarine melt. And while these are both fresh and cold water sources, their property compositions do differ ever so slightly, and this difference can be picked up in temperature and salinity space. So submarine melt is what we consider ice melange to be, and this black line is what's representing submarine melt. And this dashed line represents runoff. And this, as you move to the right of the plot, this is as they're mixing with ambient ocean water. So here you're going to get water masses with the greatest content of runoff. Here are water masses with the greatest content of submarine melt. And then as you move up, you're going to get water with a greater content of just the ambient fjord water. So what we end up seeing is that the black dots, which represent after the ice melange melt, are moved down the submarine melt line, as opposed to these blue dots, which represent before. And so this is saying that our results do have a higher content of submarine melt in them, as opposed to, say, runoff. So the next thing that this plot can help us do is determine sort of the age of this submarine melt injection, because while this could be new melt, it also could be older melt that's just mixing down in the water column from before the ice melange event happened, because we know that freshwater inputs sit at the surface. So this could just be mixing vertically downward. And the way we can tell whether or not this is the case is by comparing the surface level or the surface layer before the ice melange event to the after from 50 to 100. And if these look relatively similar, then we might say that they're due to vertical mixing. But what we end up seeing is this black notch does represent an instance where vertical mixing is likely occurring. But then here we see that these black dots from after the ice melange event are hugging the submarine line closer than these gold dots, which are being pulled slightly off of it. And this is basically telling us that while our results very likely do have some addition of vertical mixing in them, they also do show a high signal for new submarine melt as well. And with that, we're able to conclude that ice melange does lead to a cooling and a freshening of the water column of 0.34 degrees Celsius and 0.18 PSUs. And this is exciting because, of course, we might expect ice melting in a fjord to both cool and freshen the water. This is the first chance we've been able to actually measure what ice melange is really doing to the water column. Before, the influence of ice melange was really understood just through model estimates. So what this allows us to do in the future and going forward with this work is use these exact measurements as inputs into fjord circulation models. This will allow us to understand what these changes are doing to buoyancy-driven circulation and therefore heat flux up to the glacier termini. And then I also think it's worth noting that we're looking at just one instance of ice melange. But as you can see from this time lapse, ice melange is occurring throughout the summer very frequently. And each one of these times could be times where we're seeing this change occurring, which means over longer timescales like seasonal, annual, or interannual timescales, ice melange very likely has a larger impact on fjord circulation. So in summary, we've been able to see that how ice melange changes fjord water properties. And with this information, we can begin moving forward to understand how this changes not only buoyancy-driven circulation, but heat flux towards the glacier termini. And with that, we can begin understanding where ice melange fits into the conversation of ocean and ice feedbacks, or ocean and ice interactions, which ultimately affect glacier stabilization and ice sheet mass balance. Thank you. And then also, since I have a second, I just really want to thank Dave Sutherland and Cole Bibb for all their help throughout this whole process. Yeah, I mean, I couldn't have done it without you, so thank you guys.
Great job, Rachel. It looks like we do have a question in the chat for you. Does it change in space? Can we see this in other fjords?
Yes, so ice melange does occur in other fjords throughout Greenland, but how it shows up in this fjord and what it's doing and the way it's interacting with the glacier all varies. So I don't totally understand all the different dynamics of all these different fjords, but I think that's a really interesting thing to look into in the future, because yes, it does vary, but you're going to see ice melange very likely in other fjords across Greenland.
Great. Any other questions, verbal or typed, for Rachel?
I don't know if you've seen recent studies. I think recently in news we talked about how the Iceland glaciers are leaking mercury. I'm not sure that was directly related, but I was starting to see if you think that will possibly interfere with any of your results in the future.
Oh my gosh. I mean, that's a great question. I have no idea. I don't know anything about mercury, but that would be a super cool thing to look into in the future.
This isn't a question, but I do want to compliment you on how well you described and all of this very complicated information in a very accessible way. As someone who knows nothing about glacier science, I really appreciated that. So
thank you so much. That means a lot. Thank you.
We have another question in the chat, and I want, this is directed at Rachel, I believe, but I actually think there's quite a bit of insight that all of our participants could bring. So what do you think will happen with climate change? Rachel, do you want to answer this in regards to your project, and then maybe other participants can chime in as well?
Sure. Yeah, so I think that's something that everybody kind of wants to know. It's still yet to be discovered, and as climate change continues to happen, we'll start to understand what it's doing more and more. To speculate, we know, I think I would say that ice melange, ephemeral ice melange, temporary ice melange specifically, might occur more frequently. And the reason that I'm thinking this is because we know that ice melange forms from high calving activity. That's one of the materials for ice melange, and calving activity is likely going to increase in the future. We also know that ice melange relies on cold temperatures to form, and as temperatures are warming up, you're going to get that less. So you might see more instances of ephemeral ice melange that's not persistent necessarily. But then, of course, there's the other side where we know high calving activity also, like I said, provides a lot of material for ice melange, but also can help support persistent ice melange. So this could either is, yeah, it's just kind of interesting. There's a lot of things going on. So higher calving activity could result in more ephemeral ice melange. It could also help support more persistent ice melange.
Great. Bird nerds, do you have anything to add about how your project kind of added to this conversation about climate change?
Oh, go for it, Rachel.
I'll just say a quick thing and then you should add, Al. I just want to say that there are a lot of instances of, climate change impacting bird migrations in a ton of various different ways, including, the time of season and time of the year that birds are migrating. But I think with regards to working towards, all these solutions that we know of for, working to combat climate change, I think it very much so needs to be a global conversation. And I think that incorporating different perspectives and the voices of people from different parts of the world is incredibly important when talking about, working towards these solutions for climate change. And so I just wanted to toss that out there.
Yeah, I totally agree. And I was just going to say that, unfortunately, climate change is an interdisciplinary issue. So it's affecting many different disciplines. And I think that's part of the goal with environmental ed, is to start thinking about things from an early age. Not necessarily like, hi, third graders, climate change is a thing and you have to solve it because we're not doing it in time, but really kind of enforcing or encouraging more this love of nature and teaching what we've had to spend a lot of hours in the classroom learning, helping them learn that a bit in a more inherent way, I think will do wonders in the future. Obviously, we have very current and very real concerns that need to be addressed before, the kids that we teach, before they grow up. But I think creating this new generation of scientists or of of minds that are curious about nature and about the environment that they grow up in is extremely important for mitigating climate change in the future.
Yalin or Anna, do you have anything to add?
Yeah, I can say a little bit. I guess like climate change, one big concern is we're talking about how like jellyfish populations are increasing. I guess I could talk about this a little bit earlier. Is that like jellyfish are pretty resilient, like sea warming is happening as well as hypoxia and acidification, and they actually can thrive pretty well in these conditions while most fish populations can't. So they actually might be outcompeting a lot of fish populations, which is why they see so many jellyfish blooms, because like when more water happens, they actually like will increase populations while most fish species will decrease. And so you're seeing that maybe in the future they may actually outcompete like all the other fish populations if it gets like any worse, because they can thrive so well in this like hypoxia and then also acidification and even increased water temperatures.
And is there, do you know of any examples of what would happen if we had too many jellyfish in the ocean?
Yeah, as I said, they feed a lot on zooplankton. And if you looked at what zooplankton is, it's a lot more than you think, like fish larvae, as well as fish eggs, and then also crustaceans like their larvae. Like a lot of larvae is considered zooplankton. And we rely on a lot of that to be like kind of like our commercial fisheries. So we're actually that's gonna end up leading to like a human cause where it's gonna affect like kind of like what we are feeding on. And I see a question in the chat says, jellyfish diversity changing our only biomass? I'm not sure specifically on diversity. Honestly, like jellyfish, they haven't really like evolved that much over the years. Like they're pretty simple species, so they haven't really changed too much. Like I think that's why they also can like thrive so well, is that like they don't have like a lot like going for them. Like I have cut up jellyfish in the lab, like literally they're in half and they will still survive. As long as they can eat, they'll still survive. And so they're so resilient, and honestly, they haven't evolved too much. Since they're such simple species, they can really like thrive in like any condition.
Thank you. And I know you touched on this in your presentation a little bit, but do you have any goals with what your research will do in regards to helping the climate change conversation?
Yeah, so that's definitely something that I'd say I'm concerned about in terms of pollinators and plants kind of moving forward into a changing climate. I think that doing cover cropping with native plants would definitely be not a solution, but a nice kind of mediation to kind of helping insects in the surrounding landscape. But what I would really like to see in the future is more research about phenology, so like life cycle timing for plants and insects, because what I'm kind of expecting to see and what I've read a couple studies about is kind of mismatch timing. If flowers flower earlier, then bees are out and things like that. Like I'm concerned about mismatch in the future. So I think that's also why it's important to collect insects and record the occurrence data that I'm getting right now to kind of get an idea of when things are flowering and when things are active, so we can maybe predict in the future if we will see mismatches and then work on solving that, hopefully.
Great. Any other questions? We have about 14 minutes left for follow-up questions.
I'm just going to touch a little bit more on the question where it's asking, like, is jellyfish diversity changing in biomass? I guess I read that a little bit long. I guess you meant like location diversity is changing. It's not really that much because honestly, most jellyfish don't really like actively swim anywhere. Jellyfish don't have central nervous system, so they really aren't actively thinking about where they're going. They just follow the currents. So if there are like changes in the currents of water, they'll just flow with it. I think we've seen like a few changes within like jellyfish species kind of sprouting out where they aren't usually, but it's more like localized, like near the coast because of that eutrophication. They'll kind of flow in with like the water. Usually during like El Nino events, you might see a little bit more changes in diversity, but it really just depends on what like the changes in the water currents are. And there isn't like too much drastic changes on that specifically at the moment. But like it really just, they kind of just flow with the water because they aren't actively swimming anywhere most of the times.
Great. I have a question for the bird nerds. You had mentioned that you guys incorporated Indigenous teachings into your lessons for the elementary students. As a Native person whose family is not from this area, but rather the northern Midwest, I know that these can like range so drastically, the teachings. And with working with, a group that is not only talking about migration here in Oregon, but also in Indigenous lands in Mexico, how did you incorporate different wisdoms from different cultures?
I can take this question. So within our curriculum and lesson plans, we, in the introduction lesson, did a Indigenous land acknowledgement where we taught the students what the names of the tribal lands that they lived on were and what the name of the tribal land of our partners in Guanajuato. So here in Eugene is Calpuya-Alihi, and then in Mexico, Guanajuato is the Guamares Indigenous land. And so we had a field journal activity where they were learning this and working with that. And then we also incorporated Indigenous storytelling from the Blackfoot tribe about the belted kingfisher and how it got its war bonnet feathers on its head. And so it was really interesting to incorporate that in our lessons. And we also had some art with it. And it was just kind of a interdisciplinary way to teach these students about how this story has been passed down through generations and how it's a part of learning about the natural environment and birds. So I think in the future, the program hopes to maybe incorporate a little more, as Rachael was touching on earlier, of communication between here and Guanajuato, and maybe incorporating some of the Indigenous storytelling from Guamares tribes.
Fabulous. It looks like there's a follow-up for you guys as well from your fearless leader, Professor Lynch. Can you discuss the biggest challenge that you had to overcome as a team or individually?
I will say that as much as I love working with the team, it's really hard. Like 11 people, that's a lot of people to coordinate and collaborate with. And with so many layers of being online, working, communicating with students, communicating with community partners and teachers, that's a lot of coordination. And I think initially everyone getting their footing there was maybe the biggest challenge. Of course, like the virtual setting was difficult, but I think just collaborating and really getting that communication down was the most important step to be successful with teaching lessons. And then the added element of some students having started in winter term, some students having started in spring term, or kind of at different levels of engagement with the project. There's different group projects going on. So I think that would be the hardest thing, but definitely not something that wasn't able to be overcome or worked with. Yeah, that's what I would say.
I think also adapting to this virtual format was very challenging, especially because, you know, we're trying to teach kids about the environment. It's like, why can't we go outside and be with them outside? And so that was just like so challenging to try and create lesson plans that are centered around the environment and getting kids outside without being able to go outside and do environmental education outside. So I think that was a huge challenge as well. And yeah.
I will add, I think our bingo activity was one of the ways we combated that, with really trying to promote our students to get out on their own and you know, observe all the different things. And it was really cool to share our own experiences, you know, as instructors, like sharing our stories of what we saw outside. And then as the weeks went on, the kids you know, were opening up more and telling us their stories and what they saw. And it was just a very cool experience that they were still getting engaged in doing that on their own. So.
Yalin and Anna, I know you both had your opportunity to kind of get your hands on some samples, like literally there, instead of having to be fully virtual. Did the virtual society impact your research at all? And Rachael as well, Rachael Peterson, I should say. How did virtual impact you?
I don't think it impacted me too much because my lab is pretty small. It's like 7 people. So like the main issue is that we just had to correlate like who was going into lab and like alternating. So there wasn't more than 2 people ever at a time because the lab is pretty small. I think for the first time, like when COVID shut down, so I wasn't really doing anything during like spring term of last year because like none of us were on campus or anything. But I did have the opportunity to go to OIMB. And it was just me and my mentor Marco. We went, and so like we just had to. It just like was a lot less interaction. But the OIMB campus is pretty like small and like kind of like their own little bubble. So we just kind of had to create a new bubble at the OIMB campus and play with that. And so it's definitely harder because it's also because we haven't really like interacted with lab members. Like it used to be one of my favorite parts of the week to go into lab and just interact with people and like see all the samples. So I think it was just that the really main interaction side just kind of lacked that kind of like social aspect, I guess, to my research. But so it was more just kind of like lonely me at ducks by myself, you know, doing things. But overall, I don't think it interrupted it too much besides kind of the social aspect of it.
Yeah, I would agree with that. And I'd probably say for me, like being able to actively go out monthly to the hazelnut orchards and do collecting has just been like really amazing, considering everything else is online right now. So yeah, it's been super great to like be outside and actively collecting and doing field work. It definitely makes me feel a lot more connected to my research than I would if it was all online, I think. One thing I do really miss currently is when doing data processing, sometimes it's really easy and it's a lot more helpful to like sit with a couple other people in my lab and work on, like making sure you're entering data correctly and like when you're coding and doing things like that. But yeah, it's been really nice to do field work at least.
Yeah, so I'm not sure exactly how the virtual environment impacted what I'm doing, because I think that glacier science just in general, because of accessibility, is normally virtual. So the data that I was working with was already collected, and then I supplemented that with satellite images that, of course, I can get on my computer. Would have been cool to go out in the field, but probably not what I would have been doing this year anyway. So I guess one thing to what Anna was saying, I had to learn a few new programs, and I ended up just spamming the inbox of Nicole, and she was so helpful. But it definitely, I miss that like in-person aspect of it for sure, because it would have been so much easier to just like turn to the person next to me and be like, hi, can you help? So yeah.
Great. I want to pause for any other questions before I give my final question for the afternoon or for the morning.
I guess this isn't directly related to your research, Rachel, but I don't know if you've seen the news of kind of how like the melting of the glaciers is causing like the Earth's axis to like shift. I don't know if you've seen that. I just thought it was really interesting if one of you heard about it. And if I don't know if like if the ones in Greenland are really affecting that. I know it's primarily the ones in, like the North Pole, but I just wondered if that's like something you've heard about.
Yeah, I need to read the news articles that you're reading. Yeah, I haven't seen that, but that's fascinating. So I'm gonna look it up after this. That's cool.
Yeah, I don't know. It just made me think about it because like the environmental leadership program, I'm also in that. And Peg Boulay, who's like my like leader, was talking about it during one of our field days on how like it's shifting like 8 inches a year, I think. So like we aren't seeing the effects of it, but it might be affecting like our GPS coordinates system. So like since the coordinate system is based on the Earth axis, that's why like sometimes like if you look at something, it might be a little bit off from where it used to be because it's like messing with the GPS coordinates. I don't know. There's something.
Yeah, that is so interesting. Thanks for mentioning that.
Great, we have just 3 minutes left, so I want everyone to give their quickest answer if they have one of why undergraduate research symposium and what's next. Are you planning to continue this research, I guess?
To me, this is my 3rd year presenting at the symposium. I feel like this just gives us kind of like a good like real life example of like how we present research, because it's one thing to do the research, but it's another thing to communicate it. And honestly, unless you can communicate it to the audience, then you really are just doing research for yourself. You should really be doing research for like the good of everyone. So kind of like learning how to do science communication is really a vital part of doing research. And so doing so, it's kind of helping you a little bit pushed in the future. We're going to be doing a lot more science communication. And then I will be graduating end of this term, so I will be submitting my thesis next week, and then I'll be on, and then hopefully maybe I'll keep working on this in the future to work toward publication.
Yeah, I second that. This has been a great experience in distilling down my research into a 10-minute talk.
I have kind of a question in the chat that could be added to this of, there's a lot of things that we learned. For me, this was also very accessible as a non-STEM person, so thank you for that. But is there any opportunities that you can think for your research or projects to continue to be translated for diversity, equity, and inclusion purposes? Like I believe the bird nerds y'all said you had a Spanish translation team, right?
Yeah, so we've been translating all of our lesson plans and sharing them with the schools and our partners down in Guanajuato, Mexico. And we also mentioned, or at least Natalie mentioned, that we'll be working to incorporate more indigenous knowledge, especially from tribes in Mexico in the Baja Valley, and making sure to incorporate that into our teaching. And this program will definitely continue on to hopefully evolve more and incorporate more teachers and more grades in more places. So.
Well, I do want to be conscious of time. So for those of you who have an answer to that, feel free to put a message in the chat as well. I guess we don't have a chat. we'll try to get back to that maybe on your YouTube page eventually when this video is posted. But I do want to encourage everyone to think about that accessibility component as well as the giving credit where it's due as far as diversity, equity, and inclusion is going. Just to end things out, I want to say this was a fabulous panel, so engaged. I've never run out of time before in one of these, so congrats on that. You have taught me so much, and I'm sure you've taught other people as well. I also want to give a shout out to our attendees for not only being respectful, but it also looked like we had a lot of your mentors hopping in and out of this. So a shout out to them for all of the work that they helped behind the scenes for this as well. Be sure to go give a virtual high five to them as well. But anyways, have a good day. And you can unmute and clap if you want. I think you deserve it.
Yes.
Bye.
Thank you.
Thank you.
Thank you.
Thank you.